my talk is about media modulation using switchable signaling molecules. And this is joint work with many different individuals at my university. So the PhD students, Lukas, Mike, and Moritz are involved, and also 2 postdocs, Sebastian and Max, and 3 of my colleagues, namely Andreas, Heinrich, and Katrin, are involved in this. So actually, Maike is Katrin's PhD student. And I will talk a little bit more about them later in the talk. So here's an outline. So first, I want to present the general idea of media modulation, right? So what it is about in the context of molecular communications. Then we will discuss 2 switchable signaling molecules that are suitable for performing media modulation, namely FlipGFP and another GFP that is called Dreiklang. And then finally, I want to report a little bit about the status of our research on this topic, in particular as it pertains to the theoretical modeling, but also establishing an experimental setup. Now, what is media modulation? So let me motivate this a little bit. So this is the typical setup for a molecular communication system that I guess many of you have seen many times. So we have a transmitter that emits signaling molecules. Those propagate in the channel, maybe by flow and diffusion. And then there's a receiver that can detect the presence of these molecules. And here we look at a closed loop system. So, you know, there are several rounds of transmissions, maybe in a microfluidic channel or in the cardiovascular system. Now, this setup has some shortcomings that we all have worked to overcome. So one is, of course, that the transmitter has to be replenished with these signaling molecules, which might be challenging. Then the transmitter is in the channel, and this may change the propagation of the molecules, so the flow profile, for example. And also for repeated emissions, there might be intersymbol interference, right? So the channel may get soiled basically with these signaling molecules. Now in nature, the modulation process is performed often a little bit in a different manner. In particular, in epigenetics, the gene activity can be switched by methylation to an on or an off state. So basically the property of a molecule is modified in order to carry information or to convey information. Similarly, proteins, some proteins are phosphorylated and by enzymes, and then basically their status changes, and then they have different effect on receiver molecules, let's say. And in redox reactions, enzymes generate reduced reactive oxygen via an electron transfer, and this can be used, for example, to defend against pathogens. But again, basically the signaling molecule or a molecule is changed. The properties of the molecule are changed. And in this way, information is modulated and conveyed. So the idea is to basically use the same principle for synthetic molecular communications, and basically to embed the information in the state of a molecule. And this is what we call here media modulation. Now, the advantage of this is that the signaling molecules can be already in the system, and we just use them or modulate them then from outside, right? For this, they have to be switchable. So their state or their status, we have to be able to change it. And as we will see later, there are signaling molecules that are one-time switchable and others that can be reversibly switched, and both have sort of advantages and disadvantages. The switching happens basically based on an outside stimulus, right? So this could be optical or chemical or electrical. And we need a receiver, of course, that is able to differentiate between the different states of these molecules. Now, here's sort of an example setup how this could look like in a transmission system. So we have signaling molecules that can be switched between two states, a state A and a state B. And the receiver may only be able to detect molecules in state A. Then we could convey information by having a transmitter that basically switches the molecules from state B to state A if we want to convey a binary 1. And it can remain inactive if a binary 0 is to be transmitted. So this is sort of an on-off modulation. And here we have an additional unit that we call an eraser. And this eraser can switch the molecules back to the original state. So here you see the setup. So at the beginning, we have this eraser that basically deletes the memory of our system and switches all molecules to state B. Then the transmitter modulates the molecules and switches them either to state A or leaves them as they are. Then they are transported to the receiver and the receiver detects basically. Now, to see the advantage, we came up with a little simulation here, or my student Lucas came up with a little simulation example, I should say. So let's first look at the conventional system. So here we have this point source transmitter. It emits the signaling molecules. And if you have a one-time emission, then of course the molecules propagate as a little cloud in the channel. They're detected at the receiver, and this works quite well. However, if you have repeated transmissions here on the right-hand side, then you see that over time your channel gets soiled if it's a closed channel. And then in the end, you will not be able to detect reliably because there's a lot of interference from previous transmissions. Now, with this with this media modulation setup, this looks a bit different. So here, the signaling molecules are already in the environment, right? So we don't have to emit them; they're already there, and there are these red and blue dots here in my example. And then, as you can see here, on the left-hand side, the transmitter basically modulates them and switches them to state A if needed. So those are the blue dots. These blue dots then, the molecules in this state can be detected at the receiver. And if we have a closed loop system, if the molecules pass by again at the eraser, they are set back into the original state. And as a result, there's no soiling of the channel. So this is sort of intuitive. And then we also have a little simulation result here. So we see here the number of molecules counted at the receiver versus time. And wherever you have this greenish bar, this means that we want to transmit a binary 1. So we modulate, and where it's white, basically we send a 0. Then you can see at the beginning of the transmission on the left-hand side, we can see clearly for the red curve, which is the point source transmitter, we can see whenever we send a binary 1, there's basically at the receiver an impulse received. This can be used for detection. And for this media modulation, we also have sort of an impulse, and based on this impulse, we can also detect. Right. But as time goes on, right, if we go more towards the right-hand side, then we see the soiling of the channel for the conventional transmitter. And basically, we have this red curve going up and down. And there's really no relation between what we do at the transmitter and what we observe at the receiver. So we cannot basically communicate reliably. On the other hand, for the green curve, which is the media modulation, you can see still after many transmissions, we can clearly see these peaks whenever binary 1 was transmitted, and then afterwards the signal goes down again to a lower level, which means that a binary 0 was sent. Now, a short intermediate summary, right? So we saw that this media modulation may have some advantages, and we have to be able basically to switch molecules. This could happen one time, right? So one-time switchable molecules, and we will come to those in a minute. Or as in my example here, this could happen reversibly. So we can switch back and forth. And then, of course, we are resource efficient since we use the same signaling molecules many times. And we avoid indecimal interference because we can set them back to the original state. Now, all of this hinges, of course, on these signaling molecules, right? So the question is, do such signaling molecules actually exist, right? So can we actually, either, design them, or maybe some are already in the literature, right? And this is the second part now of my talk with these signaling molecules. And the answer is yes. I mean, such molecules do already exist. And an example for a one-time switchable molecule is called FlipGFP. And this FlipGFP in its original state, which we call here the B state, is non-fluorescent. And it can be switched into a fluorescent A state. And this can be done, for example, by getting into contact with an enzyme that's called caspase 3. Now, these caspase enzymes, they are markers for cell apoptosis, so of programmed cell death, basically. And they occur, for example, around tumors, so around cancer tumors, and are used for detection of cancer cells or biomarkers for cancers. And here, as I said, in state A, these molecules, these FlipGFP molecules are fluorescent. And then we can imagine a setup as shown here in this figure. So we inject these FlipGFP molecules somewhere in the cardiovascular system. They travel in our system. And then if they encounter cancer cells or these caspases, then they are converted to the state A, so to the fluorescent state. And at a different point in the body, we can observe these signaling molecules again and then decide whether they have been switched, which is an indicator for these caspases, or not. Now, how do they work, these FlipGFP? So this is illustrated here in this figure. So in its original state, in the B state, these FlipGFP molecules have a certain conformation, right? So a certain three-dimensional structure. And in this structure, they are not fluorescent, right? So that's the ground state. But then if they get in contact with these caspases, then a certain part of the protein is cut. And due to this cut, the spatial rearrangement, they rearrange spatially, right? So they change their conformation. And then in this rearranged state, right? So they change their three-dimensional structure. They become a fluorescent. And this fluorescence then can be measured by a corresponding fluorescence measurement. So those are the FlipGFP molecules, and this is non-reversible, right? So once you cut them, then it's, game over. You cannot get back to the original state. The second example that I want to present here is this green fluorescent protein variant Dreiklang, right? And Dreiklang, so that's a German name. So this was actually invented by German, chemists. And this can also assume two states, an A state and a B state. The A state is the active state. So this is the fluorescent state. And the B state is non-fluorescent. And the interesting property they have is that you can switch back and forth between the A state and the B state. And for this, you need to illuminate these GFPs with different wavelengths, right? So there's one wavelength to switch them from B to A. If you illuminate with a different wavelength, you switch them from A to B. And then there's a third wavelength that you need for the fluorescence measurement. So in total, 3 wavelengths are involved for writing, erasing, and reading. They're stable, switchable over many such cycles. So if you apply about 160 cycles, then still you get a certain reduction in the intensity of the fluorescence, but with 50% reduction, you can still do reliable measurements. And another property they have that's less desirable is that they also spontaneously switch, right? So even without an induced switching, if they're in the channel, they may still switch spontaneously from state B to state A, so from the non-fluorescent to the fluorescent state, due to thermal relaxation. So then the system can be set up as shown here, right? So this is basically what what we saw before, right? So we have a closed loop in this case, in this example. the eraser sets the molecules back into the B state. The transmitter modulates them using a light source, right? Illuminating them with a certain wavelength in order to modulate them, basically to convey a bit 1 or bit 0. And the receiver performs this fluorescence measurement with a third wavelength, and in order to decide whether a 0 or a 1 was transmitted. Right, so with this, we can realize a closed loop system. Now, in the last part of my talk, I want to show you some results that we obtained both theoretically and experimentally for this media modulation. So the setup that we analyzed is shown here. So it's very similar to what we've basically seen before. So there's an eraser, there's a transmitter, and there's a receiver. The molecules propagate by uniform flow and Brownian motion in the channel. So, the uniform flow is, of course, an approximation that we make here because our main interest is elucidating basically the properties of this media modulation. And in contrast to the conventional system, there's more randomness in the system. And here I list sort of the sources of the randomness. So first of all, the initial state of your signaling molecules is, of course, random, right? So this you don't know a priori. Then the switching from one state to the next state is not deterministic. So that's also probabilistic and has to be modeled accordingly. The distribution of the molecules in the channel is random, right? So you only can modulate those that are in the area where your transmitter is, right? So that's a random element. Then the molecule propagation is, of course, random because of the Brownian motion. There's a spontaneous switching, as I mentioned before, right? So the molecules may switch back to state A. That's a random element. And the fluorescence measurement is also random, right? So you have different sources of randomness. All of those have to be modeled accordingly. And we did this in this paper here that was published recently in TCOM. So I don't want to go too deep into this. Instead, I want to show you some effects and results. So here as an example, we consider the bit error rate as a performance measure. And we plot this here as a function of the input power density at the transmitter, right? So the power intensity of the light that you use at the transmitter for modulation. Then we also investigate the impact of the number of signaling molecules in the system and the impact of the light intensity that's used at the fluorescence, at the receiver for this fluorescence measurement. And while we see for the light intensity at the transmitter, the higher the intensity, the better, right? Because you reduce the random element and you can get a lower error rate basically by, using higher light intensity. The same is true at the receiver for the fluorescence measurement, right? So the higher the intensity that you use for the fluorescence measurement, the less randomness you get in this measurement, and therefore the bit error rate decreases. And as you would probably imagine, if you have more signaling molecules in the channel, then also the randomness decreases. So you can see here, there are some error floors in some cases, right? So even for very high light intensity at the transmitter, you still get errors. And this is simply due to the other two random effects, right? So they may limit the performance that you can achieve if you do not design the system properly. Then in the second example here, we look at the error rate as a function of the power that we use at the eraser. And here you also see that if you don't use enough power at the eraser, then you will get higher error rates. And this is because you're not able to switch all the signaling molecules back to state B, right? Back to the ground state. And as a result, you get intersymbol interference. However, if the power of the eraser is high enough, you avoid this, and then there are other factors that limit your system performance. Now, in the last figure here that I want to show you, we sort of see an interesting trade-off of the error rate versus the velocity of the liquid, right? So this V is the velocity of the water or whatever you use as the medium that goes through your tube system. And you can see that the error rate curves, they have sort of first a decreasing and then an increasing behavior. So meaning there's an optimum velocity. Now, where does this come from? Well, if your velocity is too low, right? So if you have a very low velocity, then there will be a lot of spontaneous switching in the channel because your molecules take a long time to make it from the transmitter to the receiver. And this decreases the error rate, or this increases the error rate. On the other hand, if your velocity is too high, then there will be not a lot of switching, but the problem is then that at the transmitter, your molecules will not spend enough time in the area of the transmitter, so the modulation process becomes more random, right? So you will not be able to basically switch enough molecules to get reliable transmission, and therefore there's an optimum velocity basically here. Okay, so this was the theory. Now a few words about our experiments that we have just started. So this is done in the lab of my colleague, Catherine Castiglione, and she's actually a biological and chemical engineer. And we have a joint project where we basically investigate both the theoretical and the practical implications of this media modulation. And in particular, she has done measurements, first measurements with this Dreiklang GFP. So here's sort of the setup that we have in the lab. So it's similar to what you saw in the theory, right? So we have a eraser unit, we have a transmitter unit, we have a flow cell that's used for this fluorescence measurements. And then the GFP is in these tubes. And finally, there's a pump that generates the flow, right? So here in this figure B, in the subfigure B, you see sort of the setup in the lab that we have so far. And the three results figures I will discuss a little bit more in detail in the following. So we did first measurements with this GFP. In particular, we measured the fluorescence intensity. And in this figure, what you see is the sort of the first bar is for the initial measurement. So before these GFPs have been ever switched. And you can see there the fluorescence is the highest compared to the other cases. And then the other bars are after switching cycles. So basically, we first switch the GFP off and do a fluorescence measurement, and that's the red bar. And then we switch it on again, right, using light with a different wavelength. And then this is the blue bar, basically. And you can see that over multiple switching cycles, the red bar decreases, right? So actually the red bar should be 0, right? So in the off state, we don't want to see any fluorescence. But since the switching is not perfect, there is still some residual fluorescence. But we still see enough difference between the red bar and the blue bar in order to say that we could reliably send zeros and ones, right, with this method, right? So there's still enough distinction basically between those. And those are for 5 cycles. As we said, you can do many more cycles without significant performance degradation. Then in the next figure, we measured the absorption spectrum of these GFPs. And again, the sort of green curve, I guess, or turquoise curve, this is for the initial molecule before switching. And then the blue and the red curves, those are for the on and off state. And you can see that in the off state, we have a local maximum of this spectrum at around 350 or 360 nanometer. And this is used then to switch the molecule on again, right? So you illuminate it with 365 nanometer light to switch it on. And then you see for the blue curve, which is an on state, we have a maximum at a little bit over 400 nanometers. And this is used for the off switching, right? So this frequency is then, or this wavelength is then used for the off switching. The measurement, the fluorescence measurement is done with light at about 500 nanometer. And finally, we also investigated the spontaneous switching. This is shown in this figure. And here we switched basically the molecules first off. And then we waited basically and performed measurements. So this is the orange curve here. And you can see that over time, the fluorescence increases because of this spontaneous switching due to this thermal relaxation process, right? And it takes about 20 minutes for all the molecules to be in the on state again. And therefore, how long you can transmit is of course limited in this case, right? So how long your transmission can be because of that effect. Okay, so those are our first results, and you know, we are now in the process to set up the entire transmission chain and basically to confirm some of the long-term transmission results that we got from our theory. Okay, so here's the summary basically. So basically we advocate this media modulation, and the neat property that we have here is that we can use molecules that are already in the channel. They have to be switchable, of course, but it seems that such switchable molecules exist both in nature, and also synthetic molecules have been generated or that have these properties. And of course, there might be more research to get molecules with, you know, even better properties than those that already exist. These molecules can be one-time switchable or reversibly switchable. It depends a bit on the application, which ones are preferable. And in any case, the idea is to use an external stimulus to trigger the switching. And here we used light, but of course, in nature, often chemical signals are used. So other molecules or enzymes are used to perform the switching. So, you know, this might be an interesting avenue for further investigation. What are the benefits? Well, we can reuse the same signaling molecules, right? On the one hand, for the reversible switching case, we can avoid indissoluble interference. And for the one-time switchable molecules, we can do measurements basically in areas that otherwise would not be accessible, right? By basically injecting these molecules and then getting them switched at the place that we cannot reach, right? Maybe somewhere in the cardiovascular system, and then read them out at a different location. So that's the basic idea. And possible applications include anomaly detection, as you've seen, but also long-term transmissions in closed loop systems without having to worry about soiling the environment. Okay, so I think that's the end of my talk.